Hi, this is Meilun Ma, and this is my exam to regrade. So the first question is crossing over between, which the answer would be the crossing over occurs at non-sister chromatids of homologous chromosomes at prophase 1. This question I got wrong by not reading the answers correctly and picked the one with the non-homologous chromosome. Now, crossing over only happens when the chromosomes are the same to exchange different variations of the same gene. It cannot happen between non-homologous chromosomes because then they're crossing over for different genes. It would really not work. The crossing over also only occurs in prophase 1 because metaphase 2 is when the chromosomes line up to be separated. The crossing over has to occur before metaphase, so prophase 1 is the correct answer. Now, prophase 1 is also where the spinal fibers develop, and that's where the crossing over of the chromatids begin in preparation for metaphase 1. A second question asked about the haplotype of a female parent fly. It is given that the female is phenotypically wild type, but it didn't say that it is specifically homozygous for the breed. Now, looking at the genotype, we still see that recessive trait of the variants, like black cinnabar, are being represented. So it tells us that the fly has to be heterozygous for the trait, the female fly. Now, looking at the overall gene, we can see that the gene BC relate together, and the lowercase b and capitalized C relate together, and capitalized B and lowercase c go together, which gives us the answer. We need to incorporate the capitalized B and the lowercase and the capitalized C because the female is still dominantly expressed, which gives us capitalized B, lowercase c, and lowercase b and capitalized C. The third question is finding the probability of a child does not exhibit the disease. It is given to us that the disease is a autosomal recessive disease, which means that it is not gender linked, and that only 2 of the recessive gene is needed for this disease. Now, it tells us that the father has the disease, so he has the 2 recessive genes. The mother does not have the disease, but the carrier, so he is heterozygous for the gene. Because the disease is on the recessive side, we put them in the Punnett square, and we get that there are only 50% of the chance that it would come out to 2 lowercase t. We then learned that the disease is only 40% penetrative, so only 40% of 50% is 20% that will only actually exhibit the mutation. So in order to find it as a whole, we use the 100% minus the 20% and get the 80%, which is a chance that the offspring will not exhibit the mutation, but might still contains the genetic variation for the mutation. The last question is about the mice of the population. It gives us 2 questions of an albino mice, and we have to find the total variance. So for the first question, we are given an albino mice, and it is given us a brown diluted gene with a total. Now the offspring it gives out is 15 dilute and 11 black. Because the albino mice is a given, we know it has to be 2 lowercase c. It is given that there are black mice, so we know that one of the B gene has to be a capitalized B. However, in the total of the kids, we do not see any brown mice. So that means both of the albino mice gene has to be 2 capitalized B, so that any of the offspring provided will all be black. For the diluted mice, we see that the albino mice would need a capitalized D and a lowercase d variation, because in order for the black mice to be black, it has to have a capitalized D. And in order for the dilute mice to appear, the albino mice needs a lowercase d. So the albino mice is heterozygous for the dilute variation. The second question gives us that another albino mice is crossed with a true breeding dilute, and it gives us 9 brown, 7 dilute brown, and 8 black. That's, again, the albino has to be 2 lowercase c. We see that there are 8 black mice, which tells us that one of the B has to be a capitalized B. We also see that there is brown mice, which means that one of the B in the albino mice has to be a lowercase b, so that it would be present. So the albino mice is heterozygous for the black/brown trait. We also see that there is brown dilute and regular dilute offspring, which automatically tells us that the albino mice would have to be heterozygous for the dilute gene, giving us capitalized D and lowercase d because of the dilute only has 2 lowercase d, and we need to capitalize these in order to get brown dilute and black mice. Thank you.